right, well, welcome to cells. As I'm sure since we've been talking about phospholipid bilayers and osmosis and diffusion, and we've just started talking about about systems and generally, not even about cells yet. I'm sure maybe some of you have been thinking that we're probably going to be talking about cells. And we talked earlier in the day about cells being the smallest unit of living things. A cell is the smallest unit of living things. It is the building block to multicellular organisms. We, as multicellular organisms, are all made of trillions of different cells working together for the good of the whole. And so we've been talking about macromolecules, specifically carbohydrates, lipids, and proteins, and organic molecules, which they all are. And organic molecules make up cells. The cell itself is made of phospholipid bilayer, and it contains transport proteins. And so everything we talked about, we're going to see a little bit more in depth as we learn more about the cell. And cells are really, really small. And it's important to understand why they're so small. The smaller things have a higher surface to volume ratio, which means that bigger things, all right, have more surface to absorb things and to complete tasks than they do have to keep them up, which means that the larger something is, all right, it's got more surface but less volume, which doesn't do us any good. Cells want to keep as much possible things inside the cell as possible, while also having the least maintenance possible. And it allows them the flexibility to adapt to different conditions, especially when multicellular organisms like us have so many cells. All these cells can respond differently to changing environment. So the idea that cells are small is a biological norm, number one, because the surface area to volume ratio is small, and it allows the cell to do and adapt to more things. All right. There are two major types of cells. One of them is a prokaryotic cell, and the other is a eukaryotic cell. Now, we're talking about single-celled organisms right now, but eukaryotic cells are more complex, or at least they have more compartmentalization, and they are often able to congregate and form cellular masses that end up making things like tissues, which prokaryotic cells can congregate together up here, but they actually don't form multicellular organisms per se. So prokaryotic cells and eukaryotic cells are the two major cell types, and the differences are that eukaryotic cells have little compartments inside called organelles. They have a nucleus which houses the DNA, and they're often larger than prokaryotic cells. On the other hand, prokaryotic cells, they often have more than one cell wall. They do not have a nucleus holding the DNA. The DNA is just roaming around inside. And they also don't have any compartmentalization. They don't have specific little compartments like eukaryotic cells have. And because eukaryotic cells can make up multicellular organisms, they are generally more specialized and more efficient in what they do. Not better at what they do, but more efficient. So the two major types of cells are prokaryotic cells and eukaryotic cells, and the differences are we just outlined. All right. As we go into eukaryotic cells, there are two major types of eukaryotic cells. One of them being an animal cell, and the other of them being a plant cell. Now, plant and animal cells generally have the same configuration. They generally have the same organelles, except plant and animal cells have a few different things that are not the same. One of them is that a plant cell has a cell wall surrounding the plasma membrane. So right here we have a plasma membrane, but on the outside of that we have a highly reinforced cell wall made of cellulose. Animal cells move on their own, or at least have the capability to do so. So they have these things on the outside called cilia, and they also might have these things called flagellum, which allow them to move because, remember, animal cells need to get energy from somewhere else, and they're generally moving around obtaining nutrients. Plant cells have these things called they do not have these things called centrioles. All right. What you'll see here in an animal cell are this little star-shaped thing. It's called a centriole. It's actually made of microtubules, and microtubules are little protein structures that give rise to more microtubules. But we don't need to talk really about too much of them, although the plant cells don't have them. Plant cells also have things called chloroplasts, where animal cells don't. Chloroplasts are structures, they're little organelles. They're organelles that occur inside plants that are the site of photosynthesis, which animal cells, because they don't use sunlight for energy, they use organic molecules for energy, they do not have these things called chloroplasts. So plant and animal cells differ in those two ways the most. Otherwise, plant and animal cells are very, very similar. They have Golgi apparatuses, they have nuclei, they have rough and smooth ERs, they have mitochondria, which I'm sure you've heard about. They both have lysosomes. They both have plasma membranes. So we'll talk a little bit more specifics about what each of those things are. A few terms that are important to know. The cytosol is the fluid-filled space, like all this blue, the blue in the background here, and all of the blue in the background here. This is all cytosol. Cytosol is a fluid-filled aqueous substance that basically has a bunch of dissolved substances in it, and it's called the cytosol. When we include the organelles, we call this whole thing inside the plasma membrane of a cell, this whole thing inside the plasma membrane of a cell, the cytoplasm. The cytoplasm includes organelles, whereas the cytosol does not. The nucleus is a place that houses the cell's genetic material, all the material that's going to tell the cell basically what the cell needs to do in order to survive and change and adapt. And we talked a little about organelles. Organelles are small little organs. Think of your organs, the heart, the lungs, kidneys, all of those things are, the cell has their own little organs and they're called organelles. And we know that the cell membrane is made of phospholipids, and the phospholipid bilayer is arranged in a double layer where the hydrophilic heads face outside and the hydrophobic heads face inside. We talked a little bit about membrane proteins, and membrane proteins allow things to go through the membrane that either wouldn't normally go through on their own or just need to go through the membrane at a faster or higher rate. So membrane proteins are often embedded in the phospholipid bilayer in order to transport things across. There are two major types of proteins. They're called intrinsic and extrinsic membrane proteins. This is an intrinsic membrane protein. As you can see, it spans the membrane. An extrinsic membrane protein is basically just one that's located on the surface. And extrinsic membrane proteins tend to be for signal recognition, identification with the environment. They can allow the cell to adhere and hold on to things. They can destroy pathogens and things like that. Intrinsic membrane proteins are either they're going to be for transport or they're going to be to send signals from one place to another as well. And they can also be important for energy production as well. But these are proteins which are made of polypeptides, which are made of amino acids, all put together in some sort of quaternary structure. It's important to understand. The phospholipid bilayer, again, is a layer of phospholipids with proteins embedded both in it and on the outside. These proteins are really important. We talked about things passing through the membrane and the levels at which things can pass through the membrane. Things that generally are smaller and nonpolar can pass more easily, where things that are generally bigger and charged definitely cannot pass through the membrane. And we talked about all the molecules there within. But diffusion, passive and active transport, osmosis are all important processes. Osmosis being the movement of water towards the area with less potential from an area of more potential, which is usually towards the region with more solute. And diffusion, which is the net movement of substances from an area of high concentration to low concentration. So organelles are small organs inside cells that carry out specific functions. They allow the cell to specialize. They have greater surface area for interactions within the cell. They increase the complexity of the cell and its specific function. And these things, the organelles have led to multicellularity, in that many of the organelles that occur in cells came from either budding off of the phospholipid bilayer of the plasma membrane or by the cell engulfing another cell and taking it in and actually using that We call that endosymbiosis, which is a form of cellular eating, whereby the organelles themselves used to be single-celled organisms and were taken in by the cell and then used. And we'll have to talk about that more in class. It's a really fascinating subject. Cells need to do a lot of different things. They need to process matter, they need to process energy, they need to process info, they need to reproduce, they need to communicate and respond. So everything that the cell has is designed specifically to carry out one of these functions. All right, so always keep those functions in mind. All right, we can start off with the animal cell, and we're going to get to the plant cells. Unless I say otherwise, all the structures that I'm about to describe, plant cells also have. One of the most important structures in a plant and animal cell, remember these are both eukaryotic cells, is the nucleus. The nucleus has an inner and outer membrane. It's got a double lipid bilayer. It has pores to allow things in and out. It's got the outer part is called the nuclear envelope, and in the inside we've got this area where the DNA occurs, which is called the nucleolus. We don't need to know anything more specific about that. The most important things to know are the nuclear envelope, the nuclear pores, and the function of the nucleus itself, which is to house the DNA, which is basically the cellular information for life. All right. Now, ribosomes are these crazy things. This is an example of a ribosome. As you can tell, it's definitely a protein in quaternary structure, no doubt. But ribosomes are involved. They're proteins. They're superproteins made of a substance called RNA, which we haven't talked about, but we're going to talk about a little more. That they're superproteins made of this thing called RNA, which make proteins. So ribosomes are protein-making proteins, basically. They're made of over 50 different polypeptides, and they make proteins. And they're basically the way at which DNA and RNA make protein. Proteins carry out most of the functions in your cells. Things that do things, proteins do most of those things. They are kind of like the go-getters. When the cell needs to do something, they usually send out proteins to get those things done. A lot of other things are for structural stuff and for energy stuff, but when to get things done, it's all about proteins. There are two different types of ribosomes. There are free-roaming ones and membrane-bound ones. But it's important to know that ribosomes are these superproteins that are designed to make proteins. You're going to hear a lot of things about vacuoles and vesicles. Vesicles are really a term for very small vacuoles. Vacuoles are usually larger. They usually are meant for storage, where vesicles are usually for transportation of stuff from one place to another. So now we're going to move on to the ER. It's called the Endoplasmic Reticulum. If we go back here, the ER, Endoplasmic Reticulum, is basically attached to the nucleus and is receiving information from the nucleus and processing it and transporting it out. There are two types of endoplasmic reticulum. There is the smooth ER out here. As you notice, it doesn't have any dots on it. And there's the rough ER. And it's attached to the nucleus. So really the nucleus acts as a direct communication or direct link to the ER, both rough and smooth. The rough ER has all these things on it called ribosomes. And what do ribosomes do? Yeah, if you were thinking that ribosomes make proteins, then you're right. So what do you think the rough ER does? Yes, it's involved with making proteins. Now, the smooth ER does not have proteins, so it doesn't make proteins. It actually makes things like lipids. All right, it makes phospholipids, often makes steroids. So cells that have a lot of smooth ER often make steroids. Cells that have a lot of rough ER are often involved with making proteins. So cells differ in what they make because all cells are different, right? We have eye cells, we have ear cells, we have skin cells, we have toe cells, we have sex cells, we have heart cells. All our cells are making and doing different things. So cells are going to have different ratios of all of these different organelles that I'm explaining here. All right? But it's important to understand that information travels from the inside out when it comes to the ER. So the information starts where the nucleus is, and then it travels throughout, all right, and then exits and goes to the next place. But the rough and smooth ER really make rough material. They make the basic foundational material for making stuff. And the rough ER makes proteins, and the smooth ER is usually involved in making lipids. think of carbs, we don't need to make carbs, right? Because that's what animal cells are doing. They're obtaining carbohydrates from their environment, so they don't make stuff. The cells here of your tongue have a lot of enzymes for breaking down organic material, so you can imagine that the cells of your tongue are going to have a lot of rough ER because they're making proteins, which are enzymes. All right. Again, we talked about the smooth and rough ER. Often the smooth ER is involved with detoxification of alcohol and breakdown of glycogen, along with lipid and hormone secretion and synthesis, whereas the rough ER is more about protein synthesis. If we move out from the rough and smooth ER, well, maybe not, we get to a place called the Golgi Apparatus. All right, let's see if we can have a, all right, move outward here. So we have the rough and smooth ER here. On the very outside of that is called the Golgi Apparatus. The Golgi Apparatus's main job is to take things made in the rough and smooth ER and to process them, all right, and to modify them and to change them into functional form. So you can imagine that the rough proteins made in the rough ER are actually going to be folded correctly and modified. The R groups are going to be modified in the Golgi Apparatus. There are two phases, the trans and the cis phase. The cis phase is where incoming vesicles carrying stuff from the ER are coming from, and the outbound vesicles, which are taking stuff where it needs to go after it's already been processed and modified, is called the trans phase. Okay, so the Golgi Apparatus is like the it's like the finishing factory of the cell. All right, the Golgi maturation model basically states that the Golgi is what it receives, all right, and it ceases to be what leaves it, all right. So in the cis phase, you're getting incoming vesicles that are budding with the phospholipid bilayer here, all right, and they're getting modified as they move through, and then they're leaving. So the Golgi apparatus basically becomes what it receives, all right, and it ceases to be what leaves it, all right. So it's constantly changing depending on what it's receiving, okay. And it's really for modification and finishing of important molecules for the cell. There are many different types of vacuoles in things. One of the most important vacuoles in plants is called the central vacuole. All right. Remember, all cells, whether they plant or animal cells that are eukaryotic, have a Golgi apparatus. They have a rough and smooth ER. All right. They have vesicles and vacuoles, and they have ribosomes, and they have a nucleus with all its parts. So we're talking about eukaryotic cells right now. It's important to understand. All right. Different types of vacuoles are the central vacuole in plants. Central vacuole is usually designed to house water, which keeps the cell rigid and blown up. Remember, we talked about that's what plant cells need in order to keep plant shape, is they need to be rigid. There is often a vacuole called the contractile vacuole in protists, which are single-celled eukaryotic organisms, and they use that for locomotion. Oftentimes, vacuoles can be used for storage, like poisons in animals are stored in vacuoles so they don't interrupt or disrupt other parts of the cell. They often are attractor pigments in flowers, like things that you smell, like smelling pigments, are often housed in vacuoles. And vacuoles can also house undigested material and waste, so they can do tons of things. One of the most important vesicles now in an animal cell is called the lysosome, and this is only in the animal cell. Lysosomes are involved with breaking things down. So remember, animal cells need to break down organic material in order to get energy from them. One of the major ways of breaking these things down is this organelle called the lysosome, which is at a very acidic pH. Basically makes its own proteins or enzymes, and these enzymes they're designed really to chop things up into pieces, and then either eliminate them from the rest of the cell or fuse with another part of the cell where they can be used. They also destroy unwanted organelles that aren't working well, or they destroy foreign bacteria or foreign molecules that want to be destroyed. So that's what lysosomes do. All right. Now, peroxisomes are important in eukaryotic cells because waste products are being made at all times in order for cells to function. cells make energy and they make a lot of waste products, and these waste products would be toxic to the cell if we didn't have these things called peroxisomes, which are designed to break down waste material and release it and make it easier for the cell to work. All right. Hydrogen peroxide, H2O2, is basically can be very detrimental to the cell if not changed. So remember this idea of form and function. There are two really important organelles that we haven't talked about that exist in plants and animal cells. All right. On the right here we have these things called mitochondria. Mitochondria is also often called the powerhouse of the cell. It takes organic molecules and makes energy from it, by which the cell will end up using for energy. And it only occurs, this occurs in plant and animal cells. As we move to the left here, we have something called a chloroplast. A chloroplast is an organelle that has these things called thylakoids, these little discs on it that went together. A stack of thylakoids is called granum, and many thylakoids called grana. They house the photosynthetic pigment called chlorophyll, which is going to absorb light and make energy from it. Remember that plant cells can make their own energy, whereas animal cells have to obtain it from the environment. All right. If you notice, though, the form of these things, both of them have a double membrane. All right, an inner and an outer membrane. Both of them are highly folded, which means they have a lot of surface area. And both of them are involved with making energy. So it's really important to understand that eukaryotic cells they use tons of energy, and because of that they need to make lots of energy. So any organelle that is designed to make energy or use energy in eukaryotic cells here are going to have a lot of surface area, and you can see the folding here. It's really important. All right. The important parts of mitochondria are the inner and outer membrane, and then inside of the whole thing is called the mitochondrial matrix. Okay. And then inside the inner and outer membrane is called the inner membrane space. Now for a chloroplast, we have the outer membrane, we have the inner membrane, and then inside that, this space, the cytosol of the chloroplast is called the stroma. The cytosol of the mitochondria is called the matrix, the mitochondrial matrix. The cytosol of the cell is called the cytosol. Well, these are organelles which have their own specific conditions, right? And then we have the stroma, which is the cytosol of the chloroplast. All right, both of these organelles are really important, and actually both of these cells at one point were their own cells. They were taken in by cells or engulfed and eaten, and instead of being broken down, they ended up helping the cell out by producing energy for the cell. The cells learned somehow to use these organelles or these single-celled organisms to their own benefit. That's called endosymbiosis. All right. The mitochondria is present in all eukaryotic cells, both plant and animal cells. They have an inner and outer membrane. They have their own DNA, which says a lot about where they came from. They're the place of something called cellular respiration, which is using organic molecules to make energy. They make this energy from something called glucose, which is what we've talked about pretty often. Plants make their own energy, but they also have mitochondria. They make their own glucose, and they can use their own glucose. So plants are much more efficient than animals are at making things. But we talked a lot about the cell membrane. We talked a lot about these organelles, but we didn't really talk about the skeleton of the cell. Right? All cells have a skeleton in which that keeps them shaped well, keeps them strong and rigid. One of the most important things that cells have that keeps their shape and also for transport is called a microtubule. A microtubule spans the cell, all right, and it also acts as a place for transport of material. Another thing we have is called microfilaments. All right, they're also made of protein, and they're thinner rods, but they're also meant to allow the cell to change shape. Okay, so whereas microtubules they basically act as the beams for the house. They also act as little highways to get things done. Microfilaments, on the other hand, allow the cell to change shape. They're like the muscles. They're like our muscles. So they allow us to move and be flexible. All right. Both of these things are made of proteins, and each units. Each color here is a unit, all made of polypeptides in globular shape. So it's just amazing actually how much protein makes up the cell. It really is amazing. Right? Two of the main locomotory structures for animal cells are these things called cilia and flagella. And flagella is a single long microtubule that's designed to propel the cell. Cilia, on the other hand, are many short ones, and they wave kind of like a bunch of little hands, or like the leaves on a tree shimmer in the wind. The cilia are meant for kind of an oar-like movement, like a rowboat, whereas a microtubule is like a spinning, propelling forward movement. And both of these things, cilia and flagella, are made of microtubules, which are made of proteins, which are made of polypeptides, which are made of amino acids. Probably better. We won't focus on intermediate filaments for now, but that's AP biology if you like. And that's it.